You are trying to find a comprehensive understanding of what happens with a cell when it gets infected. What is unique about your approach, and what advantages does it have compared to more traditional virology or immunology?
All the work that I do is, of course, based on decades of research in virology, immunology, cell biology. And I think that I can really add to this by adding this genome-wide or proteome-wide perspective, by not being biased by proteins you already know or that you have a scientific intuition for, but looking at things in an unbiased way. And then making observations that you probably didn't have on your radar before.
And with your group, what do you hope to accomplish in the next 10 years?
The big picture of what my lab's going to do is develop technologies to understand the interactions of viruses with their hosts. And I plan to develop technologies that are scalable, so I can do this one virus at a time and not one viral gene or one protein at a time.
Okay, and what would you say, what motivates you, what inspires you to achieve these goals in the next 10 years?
I think this moment of excitement when you find something new, which is quite rare, but it's extremely satisfying. I think that's one of the most rewarding things you can experience. And also the moment when you feel that your trainees have made progress and have really grown as scientists, that's also very rewarding.
In your PhD thesis, you have used proteomics to map the human interactome, which explains your interest in systems biology. But how did you then get interested in virus-host interactions?
This was really a serendipity. So when I started my postdoc in San Francisco, my advisor pitched an idea to me. So this was the early days of CRISPR screening. It was a fairly new technique at the time. And he said, why don't we use CRISPR-Cas9 to target not human genes, but to target viral genes of a virus that has a DNA genome just like us. And this is how it started. And since then, this idea of working in a virus host system or in a host pathogen system has really grown on me. I think it's really fascinating. And yeah, that's how it began.
You still use proteomics in your research. Why is it such an important method in your work?
Proteomics is the method of choice if you want to study and characterize all the proteins that are in a biological sample and measure, not only identify them, but also quantify them. And in that sense, it's a really fundamental technique that you can plug into many biochemical workflows and use as your ultimate readout. During my PhD, that was protein-protein interactions, but now I plan to use it to study the change in the composition of organelles in infected cells.
So viruses are millions of years old, and their evolutionary origins are still shrouded in mystery. What fascinates you about viruses?
Really their disruptive power. They're very small entities. They enter a host cell with minimal genetic information, and they have the power of completely overhauling an infected cell, hijacking its pathways, reorganizing its architecture and turning it into a viral factory. And especially this idea of reprogramming pathways that our cells already have to their own benefits, studying that really gives us a perspective of finding out the functions that our cells already carry, because the viruses have really perfected that over the course of evolution, how to hijack those functions.
Arguably the most infamous virus in recent years was and is SARS-CoV-2. What do you think is the biggest learning from this pandemic?
Scientifically, the pandemic really underlined the importance of funding basic research. I think many of the purely scientific developments, such as many of the antivirals, but most importantly, the mRNA vaccines, are really triumphs of basic science, where people have funded for decades research in really rather obscure areas, and then that has really come to fruition. At the same time, the pandemic also underlined the importance of human psychology, which many scientists maybe didn't have on their radars. So it's not only important to do the science, but then also to communicate your findings, both to the public and to policymakers, in a way that is approachable to them, that is not abstract in an ivory tower, but in a way that people can digest the information and translate the information into actionable things that we as a society need to do.